We've already spent a lot of time talking about proteins and how they do a huge variety of things in biological systems. Anything from acting as hormones to antibodies to providing structures in cells, signaling mechanism, a whole series of things. And their ability to do all of those things in living systems comes out, it's a byproduct of their structure. So what we want to talk about in this video is protein structure. And to just get a high-level appreciation for protein structure, this is a hemoglobin molecule right over here. And this hemoglobin molecule, it's made out of 4 polypeptide chains. 2 of them have 141 amino acids, 2 of them have 146 amino acids, for a total of 574 amino acids. But you see that they don't just go into some random configuration. They come into a configuration that is really good for doing what hemoglobin does, and that is being a transporter for oxygen, being a transporter for oxygen within red blood cells. So how do proteins like hemoglobin, there's many other types of proteins that do many other types of things, how do they get their structure? Well, one way to think about it is there's different layers of the structure, or there's different degrees of structure. The first degree of the structure we could call the primary structure. Primary structure. And this is really just the sequence of the amino acids. When we talk about the translation step, when we go from mRNA and we go to a ribosome and the tRNA brings the amino acids and starts linking them together, it's setting up the primary structure. The DNA, the information in DNA, that's essentially what it's coding. It's coding for, well, what order do we put the different amino acids in? So this is just the order of the amino acids. Now the next level of structure, this is just the order. This is how we form our polypeptide. But how does a polypeptide start getting bent into these shapes to be able to do the different things that it needs to do? Well, the second order of our structure, I could say the secondary structure, this is due to interactions of the peptide backbone. Due to interactions of the backbone of the peptide backbone. And I have some examples of that. Right over here. You see right over here we have a polypeptide chain. We have a bunch of amino acids that are bonded with the peptide bonds. This is a peptide bond over here. This is between the carbonyl carbon and the nitrogen, another peptide bond. Between the carbonyl carbon and the nitrogen, another peptide bond. And this chain, this polypeptide chain, you could imagine maybe it goes down here, maybe it goes around, maybe it comes back, who knows? But we see that when it comes back, we still are going from nitrogen to carbonyl carbon, peptide linkage, nitrogen, carbonyl carbon, peptide linkage. But what you see happening is from these two chains, the backbones are interacting. I actually didn't explicitly even draw the side chains. I just put an R here for the different side chains. But you see how they're interacting. Right over here, we have nitrogen. Nitrogen is electronegative. It would hog the electrons from the hydrogen. So the hydrogen is going to have a partially positive charge. Oxygen is electronegative. It's going to hog the electrons from the carbon, so it's going to have a partially negative charge. And so this hydrogen, this oxygen, they're going to be attracted to each other. This is a hydrogen bond, our good old friend, the hydrogen bond. Same thing is going to happen over here. Same thing is going to happen over here. And so these two chains, these can form kind of this sheet. In fact, this is called a beta-pleated sheet. Beta-pleated sheet. Now over here I've also constructed a beta-pleated sheet, but you might notice a difference. This one went from the nitrogen to the alpha carbon. This is the alpha carbon over here, to the carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon. And this one was also going in the same direction. Nitrogen, alpha carbon, carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon. So this is a parallel beta-pleated sheet. Both of these side chains that are interacting, both of these backbones that are interacting are going in the same direction. So we would just call this one parallel beta pleated sheet. Now this one, they're parallel, but what we see going on, we go nitrogen, alpha carbon, carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon. That's on the left side, but on the right side, we're going carbonyl carbon, alpha carbon, nitrogen, carbonyl carbon, alpha carbon, nitrogen. We're going in the opposite direction. In fact, to even construct this, I copied and pasted this, but I rotated it around. You can see that I've actually drawn it upside down. And so here we have these two things. You still have the hydrogen bonds between these partially positive ends of these of this bond, this nitrogen hydrogen bond with at the hydrogen end, and this partially negative charge at the oxygen. You still have these hydrogen bonds, and these backbones are parallel, but they're going in different directions. They are, so we would say these are anti-parallel beta pleated sheets. So anti-parallel beta pleated sheets. So this is another form of a secondary structure. Now this over here, we see that the backbone is going in this. It's going in this helical structure, and we have essentially hydrogen bonds between the different layers of the helices, or between the different layers of the helix, I should say. So over here, this oxygen is partially negative charge. This hydrogen, partially positive charge. So I have a hydrogen bond. I could have a hydrogen bond over here. And so that's what gives this a helical structure. And we would call this an alpha helix. So these interactions between the backbone, the peptide backbone, that's the secondary structure. That's the secondary structure of a protein. Now, we're not done yet, because you could imagine these side chains have something to say. Some of these side chains are hydrophobic, so they would want to kind of pull that part into kind of away from, if it's in water, away from the outside. Some of these side chains might form hydrogen bonds with other side chains, so those would interact in certain ways. You could have side chains that form actually disulfide bonds, actually covalent bonds with other side chains. And we're going to go into a lot more detail of that in a future video. But the third, I guess, the third form of structure, and we'll call that the tertiary structure. The tertiary structure. This is due to interactions of side chains. So due to side chain interactions. Due to side chain interactions. And so you could imagine, if, let's say I have this, this thing over here, maybe I have a bunch of hydrophobic side chains right over here. I'm just drawing them as R, but let's just assume they're hydrophobic, and let's say that the H2O is out here. They might, and we'd have to think in 3 dimensions, but they might want to get away from the H2O. And likewise, you could maybe have hydrophilic side chains, maybe polar side chains might be on the outside. You might have a situation where one side chain, let's call it R1, and another side chain R2, maybe they form hydrogen bonds with each other. Maybe they have an ionic bond with each other. So there's a bunch of different types of side chain interactions that we could actually think about. And we'll go into more depth in that in a future video. Now, any protein that's made up of a single polypeptide is only going to have primary structure, secondary structure, and tertiary structure. But if we're dealing with something like hemoglobin that's made up of more than one polypeptide, then we're going to talk about quaternary structure. So quaternary structure. And this is all about how the different polypeptide chains come together to form the larger complex. So multiple, I guess you could say, interactions or arrangement of multiple chains together. So arrangement of multiple, in the case of hemoglobin we had 4, multiple peptide chains.